Hello everyone. Today we will be discussing enzymes, fascinating molecules that play a crucial role in the biochemical processes of living organisms. Enzymes are biological catalysts, which means they speed up the rate of chemical reactions without being consumed or altered in the process. Enzymes significantly increase the rate of reactions. In fact, in the absence of an enzyme, certain reactions may barely proceed or might take a long time. However, when an enzyme is present, the reaction rate can increase up to 10 to the power of 7 fold. That's a huge increase. The presence of enzymes can make reactions occur at a much faster pace and under milder conditions compared to when no enzyme is present. One of the key advantages of enzyme catalyzed reactions is that they happen under relatively mild conditions. Unlike many chemical reactions which might require high temperatures or extreme pressure, enzyme catalyzed reactions can occur at temperatures well below 100 degrees centigrade at normal atmospheric pressure and at a neutral pH. This is incredibly important for maintaining the delicate balance of life processes in organisms. Enzymes are also highly specific. This means that each enzyme typically acts on only one or few related substrates. The specificity ensures that only the right reactions are catalyzed, leading to the formation of the correct products. Think of enzymes as lock and key models, where only a specific key or substrates fits the enzyme's active site. Enzyme activity is not constant. It can be regulated. The enzyme's activity can change in response to the concentration of substrates or other molecules present in the system. This regulation allows the cell to adjust enzyme activity as needed, ensuring efficient use of resources and coordination of various metabolic processes. Almost all enzymes are proteins, and their ability to catalyze reactions depend on their three-dimensional structure. However, it's also important to note that a few RNA molecules have been identified to have catalytic activity as well, which is a fascinating exception to the general rule. Let us quickly understand the process of enzyme immobilization. Enzyme immobilization, an important technique used in biocatalysis, biotechnology, and industrial applications. Immobilizing enzymes involves confining or localizing them in a specific area, usually on a solid support, without losing their catalytic activity. This technique has numerous advantages, such as the ability to reuse enzymes and increase their stability in different environmental conditions. Why immobilize enzyme? Why immobilize enzyme? Before we dive into the methods, let's briefly look at why immobilizing enzyme is so beneficial. When enzymes are immobilized, they can be reused multiple times, which significantly reduces operational costs. They also become more stable, making them resistant to changes in temperature, pH, and other environmental factors. This stability is crucial for many industrial applications where enzymes are used repeatedly. And what are the methods of enzyme immobilization? There are several methods of immobilization of enzymes, each with its own set of advantages and disadvantages. Let's take a closer look at the 5 main techniques which are used in enzyme immobilization technique are physical adsorption, covalent binding, entrapment, cross-linking, and affinity binding. We will explore each in detail. Physical adsorption. Let's start with physical adsorption. In this method, enzymes are adsorbed onto a solid support such as polymers, glass beads, or activated carbon via non-covalent interactions like hydrophobic, electrostatic, or van der Waals forces. This is one of the simplest and most cost-effective methods of immobilization. What are the advantages? The advantages of physical adsorption method is it's straightforward and inexpensive, making it a popular choice. Of course, this method also has its own disadvantages. The disadvantages of physical adsorption are enzymes may leach out under harsh conditions, and the absorption process can sometimes negatively affect the enzyme's activity, especially if the forces used to bind the enzyme are too weak or disrupt the enzyme's structure. Next, we have covalent binding. This method involves chemically bonding the enzyme to a support material. The covalent binding is nothing but sharing pair of electrons. Like a resin or polymer, through functional groups such as amines, carboxyls or hydroxyls, the covalent bond provides a stronger attachment compared to physical adsorption, leading to greater stability. It also has some of the advantages and disadvantages. Advantages are stronger enzyme attachment and improved stability. The disadvantages are this method is more complex and can sometimes cause the loss of enzyme activity due to the binding process, especially if the reaction conditions are not carefully controlled. The next method is entrapment. In this method, enzymes are physically confined within a matrix such as a gel or microcapsules without being chemically bonded onto it. This process traps the enzyme inside a polymer or gel matrix. It also has some of the advantages and disadvantages. The advantages are the enzyme is well protected from external environmental changes, which increases its stability. The disadvantages are the major downside here is that the diffusion of the substrate and product may be hindered by the matrix, therefore making the reaction less efficient in some cases. Another technique is cross-linking, where enzyme molecules are chemically cross-linked together using bifunctional reagents. This forms an enzyme aggregate that is immobilized on a support. Cross-linking can create a very stable enzyme preparation. The advantages of this method results in a robust enzyme preparation that can withstand harsh conditions. And it also has the disadvantages. There is a risk of enzyme deactivation during the cross-linking process, and it can potentially block or alter the enzyme's active site, which may affect its catalytic activity. Finally, we have affinity binding. In this method, enzymes are immobilized via an affinity interaction using specific ligand or antibodies that bind to the enzymes. And this technique is highly selective. This technique also has some of the advantages and disadvantages. The advantages are highly specific and selective for particular enzymes. The disadvantages are they are expensive and complex to set up. And this is the process of enzyme immobilization. The enzyme immobilization is a process of confining or localizing them in a specific area, usually on a solid support, without losing their catalytic activity. And this technique has advantages such as ability to reuse enzymes and increase their stability in different environmental conditions. So these are the methods which I have explained, and also these are the techniques which are commercially used or applied for applications in enzyme mobilization. Like I said, the physical adsorption. In the physical adsorption, so adsorption is mainly can be an covalent interactions or non-covalent interaction, where you can see just an adsorption process, where the static process or dynamic process or some of the reactor loading process. Of course, this has is a straightforward and inexpensive process. Of course, it makes more popular choice, and the main disadvantage, like I said, it may leach out under harsh conditions, and the adsorption process can sometimes negatively affect the enzyme activity. The enzyme are too weak or disrupt the enzyme's structure. We have irreversible type of enzyme mobilization. So in this, the covalent bonding. The covalent bonding the enzyme to a support material like a resin or polymer through functional groups such as amines, carboxyls or hydroxyls. The covalent bond provides a stronger attachment compared to physical adsorption, leading to greater stability. Like I said, it also has its own advantages: stronger enzyme attachment and improved stability. And this method is more complex and sometimes cause of loss of enzyme activity during the binding process. This is most common. So therefore, we have another technique. In another technique, we have entrapment method. So in the entrapment method, this method entrapment method, the enzymes are physically confined with a matrix such as a gel or a microcapsules without being chemically bonded to it. This process essentially traps the enzyme inside a polymer or gel matrix. So like I said, it also has some of the advantages and disadvantages. The enzyme is well protected from external environmental changes which increase its stability. And the major downside here is that the diffusion of substrate and products may be hindered by the matrix. Therefore, it makes less efficient in some cases. And the cross-linking method, this cross-linking method where enzyme molecules are chemically cross-linked together using bifunctional reagents. This forms an enzyme aggregate, so that is immobilized on a support. The cross-linking can create a very stable enzyme preparation. Of course, this method also has advantages. This method results in a robust enzyme preparation that can withstand harsh conditions. Disadvantages are there is a risk of enzyme deactivation during the cross-linking process, and it can potentially block or alter the enzyme's active site, which may affect its catalytic activity. And this is another overview of how a particular enzyme mobilization occurs here, where E represents an enzyme molecule, where you can see an electron rich, and there is also a sharing an electrons, where you can see the E, that is a solid porous support, and porous polymeric matrix, where you can see there is a cross-linking and also a semi-permeable membrane. So all the enzymes are kept inside the semi-permeable membrane. So this is the method of the enzyme immobilization. Like I said, the carrier-based mechanism, covalent binding, adsorption, encapsulation, and entrapment, porous carrier, and non-porous carrier are some of the examples of the carrier-based immobilization technique. And this is one example of immobilized enzyme, how it generally looks after the immobilization, where you can see the enzymes are trapped inside a bead, a sodium alginate bead, and this helps an enzyme to be present and intact inside the beads. The bead will have certain porous material. This helps in the movement of substrates and also reactant. So this is one method of immobilization of enzyme using bead. And like I said, the adsorption method. So in the adsorption method, where you can see the physical adsorption, that's adsorbed onto a solid support such as polymers, and the physical adsorption such as glass beads or activated carbon via non-covalent interactions. And here is the covalent bonding, and we have metal organic frameworks. And this type of the method is generally considered to be irreversible. The covalent bonding is an irreversible method of enzyme immobilization. And these are some of the various representations or illustrations of immobilization technique, where we have a solid support adsorption method, an encapsulation method where semi-permeable membrane is trapped, as well as the cross-linking of each enzymes, and there is a covalent bonding. The solid support will help in the formation of a covalent bonding, and also an entrapment method. And this is one technique generally commercially is used in a reactors with immobilized enzymes on membranes, where this is an immobilized enzyme on membrane. When the substrates which is being added into this particular enzyme, then adsorption, or this method can be an adsorption, entrapment, covalent link, or cross-linking. The biomolecules or the small molecules or organic pollutants or small molecular compound, so that can be used for synthesis of some of the products such as polypeptides, cyclodextrins, low molecular weight proteins, small molecule sugar, fatty acids, and triolene, glycerine, etc. Or it also can be used in small molecule non-polluting, conversion of small molecule organic pollutant into small molecule of non-polluting substance, the conversion of small molecule compound into the malic acid, methanol, isomalto-oligosaccharides, and also CLI, lactose, etc. So this method, the conversion of substrates to product, can happen in a reactor with an immobilized enzymes on membranes. This is another overview of the enzyme mobilization technique. Okay, so now we'll talk about the applications of immobilized enzymes, and it has a wide range of applications. Let's look at some of the key areas where enzyme mobilization is used. This one is industrial processes. So enzymes are used in the production of biofuels, food processing, and pharmaceuticals. Immobilizations allows for efficient, continuous production process where enzymes can be reused multiple times. This also can be used in waste treatment. Immobilized enzymes are also used in bioremediation processes where they degrade pollutants in wastewater or contaminated soils. Applications of immobilized enzymes in diagnostics. They are used in biosensors to detect specific substances by catalyzing reactions, making them ideal for medical testing and environmental monitoring.